Okay, so are you ready to dive into some serious AI today?
Always.
You sent us this paper about Evo2,
Yeah.
this new model that's really changing things in biology.
Yeah. It's pretty wild.
And honestly, it's mind-blowing.
Yeah. Evo2 is uh, what researchers are calling a biological foundation model. And to get to that level,
Okay.
it's trained on a massive data set of DNA 9.3 trillion base pairs.
Whoa. Okay, so to put that in perspective for our listeners, is that like all the libraries in the world, but like in DNA form?
Even bigger. It's actually more data than all the text on the internet.
Wow.
And the crazy thing is Evo2 can learn directly from this raw DNA data without needing specific instructions for each task.
So no hand-holding necessary. This AI is basically a self-taught biology whiz kid.
Exactly. Imagine trying to learn a language just by reading a dictionary
Oh, wow.
with no grammar explanations.
Right.
That's kind of what Evo2 is doing with DNA, and it's doing it remarkably well.
Okay, I am intrigued, but let's back up a bit.
Sure.
What kind of DNA is Evo2 actually learning from?
They didn't limit it at all.
Okay.
Evo2 was trained on DNA from all domains of life: bacteria, archaea, eukaryotes, viruses.
Wow.
It's getting a truly comprehensive view of the building blocks of life.
So it's not just focused on human DNA, it's looking at the entire spectrum of life on Earth.
Exactly.
Yeah.
And because it's trained on such a diverse data set, it can make connections and predictions about biological systems that wouldn't be possible with a narrower focus.
That makes a lot of sense.
Yeah,
But DNA is notoriously complex and long.
it is.
How does Evo2 actually handle all that information?
That's where its unique architecture comes in.
Okay.
They use something called striped hyena 2, which is specifically designed to handle the length and complexity of DNA sequences.
Hmm.
Think of it like a specialized lens for a microscope, allowing Evo2 to see patterns and relationships in DNA that other AI models might miss.
So Striped Hyena 2 is helping Evo2 make sense of this massive DNA library.
Exactly. And one of the most impressive things it can do is predict the effects of mutations.
Mutations, like those tiny changes in DNA that can sometimes cause diseases.
Exactly. Evo2 can look at a mutation and predict how it will affect a protein, an RNA molecule,
Mm-hmm.
or even the overall fitness of an organism.
Wow.
They tested it on the ClinVar dataset, a huge database of human genetic variants, and the results were really impressive.
So, if I understand correctly, this could be huge for personalized medicine.
Absolutely. Imagine being able to tailor medical treatments to an individual's specific genetic makeup based on Evo2's predictions.
That would be incredible.
Yeah,
But Evo2 can also predict the effects of mutations in non-coding regions of DNA, right?
right.
Wasn't that stuff once considered junk DNA?
That's a common misconception.
Okay.
We're learning that non-coding DNA plays a critical role in regulating gene expression.
Okay.
It's not junk at all. And Evo2 is helping us understand how these often overlooked regions of DNA influence our biology.
It sounds like this AI is helping to rewrite the textbooks on DNA.
It really is.
But let's get specific. What kind of insights is Evo2 providing about non-coding regions?
Well, for one, it's showing us just how information dense these non-coding regions are.
Okay.
They contain a wealth of regulatory elements that control when and where genes are turned on and off.
So it's like a complex control panel fine-tuning the activity of our genes.
A perfect analogy. Evo2 is helping us decipher the intricate language of this control panel, revealing how these regulatory elements work together to orchestrate the symphony of life.
That's a beautiful way to put it. It sounds like Evo2 is shedding light on some of the most fundamental mysteries of biology.
It is.
But there's more, right?
Much more. It can even identify exons and introns, the
Ooh.
fundamental building blocks of genes,
Ah.
directly from the DNA sequence.
That's incredible. It's like Evo2 is learning the grammar of the genetic code.
Yeah.
Being able to distinguish the nouns from the verbs.
Precisely. And this ability to identify exons and introns has huge implications for understanding gene structure and function.
Okay, I'm completely captivated. What else can this amazing AI do?
Get ready for this because it's pretty wild.
Okay.
Evo2 can actually generate entirely new genomes.
Wait, hold on. Generate? As in design entire genomes from scratch?
It-
Is that even possible?
It is, and Evo2 is doing it.
Wow.
They've already shown that it can generate realistic human mitochondrial genomes,
Wow.
minimal bacterial genomes, and even yeast chromosomes.
So we're not just talking about understanding existing life, but potentially creating new life forms.
Right.
This is starting to sound like science fiction.
It's definitely pushing the boundaries of what we thought was possible in biology, and it raises all sorts of fascinating questions about the nature of life and our ability to shape it.
I can only imagine. But before we dive into those deeper philosophical waters, can you walk me through how Evo2 actually designs these new genomes?
It's a process called unconstrained autoregressive generation.
Okay.
It starts with a short prompt, like a snippet of DNA, and then Evo2 predicts the next base pair and the next, building an entire genome one piece at a time.
So it's like giving Evo2 a starting note and then letting it compose an entire symphony based on all the musical rules it's learned.
That's a great way to put it. And the results are just as impressive. The genomes Evo2 creates contain the expected genes, regulatory elements, even structural features, just like you'd find in a naturally occurring genome.
So these aren't just random strings of DNA.
No.
They actually resemble real functional genomes.
Right?
How can they be sure of that?
They used AlphaFold, another incredible AI, to predict the 3D structures of the proteins these generated genomes would produce.
Oh, wow.
And get this, the predicting structures were incredibly similar to the structures of naturally occurring proteins.
Wait, they used one AI to analyze the output of another AI?
Yeah,
That's AI inception.
it is.
And it worked.
It did. Which tells us Evo2 isn't just copying what it's seen. It's actually learning something fundamental about how DNA dictates protein structure.
Yeah.
This is huge for fields like drug discovery, where designing proteins with specific shapes is essential.
So we could potentially use Evo2 to create custom-designed proteins for specific medical treatments. That's
Yeah.
incredible. But how do we even begin to understand what Evo2 is actually learning from all this data? It feels like magic.
It does feel like magic, but there's a method to it. Researchers used a technique called sparse autoencoders to peek under the hood and see what Evo2 is focusing on.
Sparse autoencoders. Break that down for me.
Okay,
What are those?
think of them like super sleuths for data.
Okay.
They can sift through mountains of information and pinpoint the most important features. In this case, they revealed the specific patterns and features in the DNA that Evo2 uses to make its predictions.
So what kind of clues did these autoencoders uncover? What is Evo2 paying attention to?
It's picking up on things at all levels, from the smallest building blocks to larger organizational patterns. For example, it can pinpoint open reading frames. Those are the DNA segments that directly code for proteins.
Okay.
It's recognizing the genes that are essential for a cell to function.
So it's like Evo2 is learning the basic vocabulary of life.
Identifying the key components that make a cell tick.
Exactly. But it goes beyond just recognizing individual parts. Evo2 is also picking up on structural elements within proteins, things like alpha helices and beta sheets.
Wait, so it's understanding not just the DNA sequence, but how that sequence translates into the 3D shape of a protein?
It is. It's grasping the link between the blueprint and the final product, between genotype and phenotype, as biologists would say. This deep understanding is what allows it to generate those realistic genomes we talked about.
This is mind-blowing. It's like Evo2 is developing this deep intuition about the rules of biology. What other aha moments came from these sparse autoencoders?
Well, one fascinating finding was that Evo2 can identify prophage regions in bacterial genomes.
Prophage regions, those are remnants of ancient viral infections, right? Like finding fossils in the DNA.
Exactly. These viral sequences have integrated themselves into the bacteria's DNA over millions of years, and Evo2 is able to pick them out. So it's not just learning about the present, it's also uncovering clues about the evolutionary history of life on Earth.
It is, and get this, it's even picking up on the subtle differences in codon tables between different organisms.
Codon tables, those are like the translation guides that cells use to turn DNA into protein, right?
Precisely. And each organism can have slightly different versions. So Evo2 is essentially learning the dialects of life, how different organisms have evolved slightly different ways of speaking the language of DNA.
It's like Evo2 is becoming fluent in all the dialects of life.
Yeah.
This is incredible. But let's go back to that genome generation ability. We were talking about how it builds a genome base pair by base pair, but how does it know when a genome is complete? When to stop?
That's where things get really fascinating. It uses a concept called perplexity.
Okay.
Essentially, as Evo2 generates each base pair, it's also calculating the probability of that base pair occurring in that specific sequence.
So the lower the perplexity, the more confident Evo2 is that it's on the right track.
Exactly. And when the perplexity drops below a certain threshold, it signals to Evo2 that the genome is likely complete. It's like reaching the final punctuation mark in a sentence.
That's a clever way to control the generation process. It sounds like Evo2 is not just blindly spitting out random DNA sequences.
Yeah.
It's really thinking about what it's creating, making sure it makes sense in the context of the complete genome. But are these genomes actually functional? Could they support life?
That's the million-dollar question, and it's one that researchers are actively trying to answer. They're using a variety of techniques to assess the functionality of these generated genomes, both computationally and experimentally.
So it's not just about creating a genome that looks right on paper. It
No,
has to actually work in a living cell.
exactly. It's a huge challenge, but the potential rewards are enormous.
Yeah.
If we can successfully generate functional genomes, it would open up incredible possibilities for bioengineering medicine and even our understanding of the origins of life.
Okay, my head is spinning. This is truly groundbreaking stuff. But I have to ask, with all this talk about creating new life forms, are there any ethical concerns surrounding this technology?
That's a crucial question, and one that the scientific community is taking very seriously.
Okay.
The ability to design new biological systems raises profound ethical questions about biosafety, unintended consequences, and even the very definition of life.
It sounds like we need a whole separate deep dive just to explore the ethical implications of Evo2. But for now, can we shift gears a bit and talk about another fascinating aspect of this technology, its ability to design epigenomic patterns?
Absolutely. Epigenetics is all about those modifications to DNA that can affect how genes are expressed without changing the underlying sequence itself.
It's like adding sticky notes to the DNA, highlighting certain sections, or making them easier to read, right?
A perfect analogy.
Yeah.
And Evo2 can actually design these epigenetic patterns using a technique called inference time search.
Okay.
Essentially, it guides Evo2 towards creating DNA sequences with specific patterns of open and closed chromatin.
Open and closed chromatin. Is that related to how accessible the DNA is to the cell's machinery?
Exactly. Open chromatin is like an open book, easily accessible for the cell to read and transcribe. Closed chromatin is more like a closed book, tightly packed away and less accessible.
So Evo2 can manipulate these patterns of accessibility. How does it do that?
It leverages the power of multiple AI models working together.
Mm.
They used a combination of Informer and Borzoi, two sophisticated AI models that specialize in predicting chromatin accessibility from DNA sequence.
So it's like a team of AI experts, each with its own unique skill set, collaborating to guide Evo2 towards creating specific epigenomic patterns.
Exactly, and they demonstrated this capability in a truly mind-blowing way.
Okay, I'm on the edge of my seat. What did they do?
They used Evo2 to encode messages in Morse code, using patterns of open and closed chromatin.
Wait, Morse code?
Yeah.
Like dots and dashes?
Yeah.
How is that even possible?
They designed a specific sequence of open and closed chromatin regions that corresponded to the dots and dashes of Morse code, and Evo2 was able to generate DNA sequences that matched those patterns.
So they basically turned DNA into a biological telegraph. That's incredible.
It really showcases the remarkable precision and control that Evo2 offers in the realm of epigenomic design.
This is truly revolutionary stuff. But I have a feeling we're only scratching the surface here, right?
Yeah, definitely.
What else can Evo2 do?
You're absolutely right. There's so much more to explore. We haven't even touched on Evo2's ability to predict the 3D structure of RNA molecules and its potential to unlock the secrets of the RNA world.
Okay, I'm ready for another layer of mind-blowing. RNA world, here we come. But before we dive into that, I'm curious about something. We've talked about all the amazing things that Evo2 can do, but I'm wondering about the how. What were some of the challenges the researchers faced in developing this groundbreaking model? It does seem like there must have been some serious hurdles to overcome, especially when you consider the sheer amount of data Evo2 is processing. We're talking trillions of DNA base pairs. What kind of computing power did it take to train this model?
You're absolutely right. The computational demands were huge. They needed cutting-edge hardware, powerful GPUs, like the ones you find in high-end gaming computers.
Oh, wow.
But in this case, they were crunching DNA data instead of rendering graphics.
So they basically built a supercomputer specifically for Evo2.
Pretty much. And even with all that power, it still took a considerable amount of time and energy to train the model.
Wow.
But they didn't stop there.
Okay.
They also used a technique called distributed computing, which allowed them to spread the workload across multiple machines, working in parallel to speed things up.
So it's like having a team of supercomputers all working together on the same problem.
Exactly. That kind of collaborative computing power is essential when you're dealing with data sets as massive as the one Evo2 was trained on.
That makes a lot of sense. But I imagine the challenges went beyond just the computational side of things. Biology is messy and complex, and DNA data can be very diverse. How did they account for that?
That was a big hurdle.
Yeah.
Remember, Evo2 is trained on DNA from all domains of life:
Right.
bacteria, viruses, plants, animals.
Yeah.
Each of these organisms has its own unique genome-sized structure and evolutionary history.
So it's not just a matter of dumping all the DNA data into the model and hoping for the best.
Definitely not. They had to carefully curate and pre-process the data,
Right.
making sure it was clean, consistent, and organized in a way that Evo2 could understand. It involved a lot of trial and error experimentation and close collaboration between biologists and computer scientists.
It sounds like a truly interdisciplinary effort, bringing together expertise from different fields to solve a really complex problem. And they were able to overcome these challenges and create Evo2, this revolutionary model that's already making waves in the scientific community.
They did, and they've been incredibly generous in sharing their work with the world. They made Evo2 open source, which means anyone can access the model, the code, the data, and contribute to its development.
That's fantastic. It speaks to a real spirit of collaboration and a desire to accelerate scientific progress. So what's next for Evo2? What are the researchers hoping to achieve with this technology in the future?
Evo2 is still in its early stages, but the potential applications are vast.
Yeah.
They're constantly working to improve its accuracy, its ability to handle even more complex biological data, and its potential to design even more sophisticated biological systems.
Can you give me some specific examples? What are some of the areas they're focusing on?
Well, in human health, they're exploring how Evo2 could be used to develop more precise diagnostic tools. Imagine being able to detect diseases earlier and more accurately, leading to more effective treatments.
That would be a game changer for medicine.
Absolutely. They're also looking at personalized therapies, tailoring treatments to an individual's unique genetic makeup based on Evo2's predictions, and potentially even using Evo2 to develop strategies to prevent genetic diseases altogether.
So we're talking about using AI to revolutionize healthcare at a fundamental level. That's incredible. But what about applications beyond human health?
The possibilities are really limitless. They're exploring how Evo2 could be used to design new biomaterials with unique properties like stronger, more sustainable building materials inspired by nature. They're also looking at biofuels, developing more efficient and environmentally friendly ways to generate energy.
So we could use Evo2 to engineer a more sustainable future.
Exactly. And they're even exploring the possibility of using Evo2 to design synthetic organisms that can perform specific tasks. Imagine organisms that can clean up pollutants or produce valuable chemicals, all powered by the code of life that Evo2 is helping us understand.
It's mind-boggling to think about the potential impact of this technology. It sounds like Evo2 could touch almost every aspect of our lives.
It really could.
Oh,
And beyond the practical applications, there's the fundamental scientific knowledge that Evo2 is helping us unlock. Researchers are hoping to use it to deepen our understanding of the basic principles of life, explore the origins of life itself, and even search for life beyond Earth.
wow, from designing new materials to searching for extraterrestrial life, Evo2 seems to be pushing the boundaries of what we thought was possible in biology. It's really inspiring to think about.
It is, and I think it's important to remember that we're just at the beginning of this journey. Evo2 is a powerful tool, but it's still evolving, just like the field of biology itself.
That's a great point. So for our listeners who are just starting to wrap their heads around Evo2, what's the one key takeaway you'd want them to remember?
I think the most important thing to understand about Evo2 is that it's not just a powerful AI. It's a powerful tool for understanding life itself. It's helping us unlock the secrets of the genetic code, explore the vast diversity of life on Earth, and even imagine the possibilities of creating new forms of life. It's a testament to the incredible power of human ingenuity and the boundless potential of scientific discovery.
Beautifully said. It sounds like Evo2 is more than just a technological advancement. It's a reflection of our own innate curiosity and our desire to understand the world around us. It's an invitation to explore the mysteries of life and to imagine a future where the boundaries of what's possible are constantly being pushed further. And on that note, I think it's time to wrap up our deep dive into Evo2. Thank you so much for joining us on this incredible journey of exploration.